Today we are going to be looking at the second part of meiosis. We saw that last time we saw the introductory part of meiosis and we stopped on the human karyotype. So we are going to go into detail of meiosis today, and then we also do some questions concerning about meiosis. Some of the questions which tend to be complex to some students when it comes to meiosis. Don't forget that meiosis is one of the topics which are very easy to understand. You can grasp it, but the problem is when it comes to the questions, uh, you tend to lose the marks. So we have to solve that problem today so that we are able to uh, get marks when it comes to meiosis. All right. Uh, don't forget that we say that in meiosis, we have a process of mitosis. Should we talk about mitosis in meiosis? Just to compare the two, or the two phases, whereby you know that one of mitosis, we say that you have one cell which divides to form two cells, of which these two cells are genetically identical to the mother cell. Yes. So of which meiosis, we say that you have one cell divides to form four cells. Yes, whereby each cell is different from each other and has half the chromosome number to the parent. So those are, we have to differentiate between meiosis and mitosis so that we are able to know their importance. All right. Mitosis occurs in every living organisms. Every part of the body, we have mitosis. That's why we say that it occurs in the somatic body, in the somatic cells, which we call the body cells. And then we are saying that why do we need to have mitosis? Basically, to replace those damaged cells, to generate new cells. Yeah, that's why they are genetically identical. If you have a wound and then the cells which are being replaced are different, then that part where you had a wound is going to be looking different. So that's why you have to be with something which is identical. So two identical cells are produced with identical number of chromosomes, and these chromosomes, they're exactly the same as the parent. So mitosis has only one cycle. It doesn't have mitosis 1 and mitosis 2. No. So now we are busy generating the difference between meiosis and mitosis. Why? Because now we see that two cycles in meiosis, one cycle in meiosis, in mitosis, whereby in actual sense we can say that meiosis has two nuclear divisions, while mitosis has one nuclear division, meaning that meiosis has meiosis 1 and meiosis 2, while mitosis has meiosis doesn't have meiosis 1, doesn't have meiosis 2. Yes. So when we go to the next part, the process of meiosis, what are you supposed to look at when it comes to this? In the process of meiosis, we need to look at these four cells which are formed. These cells have half the chromosome number to the parent, and it has different phases. These different phases are the one which constitute meiosis to be a full phase. But there is a phase called interphase. Sometimes some people they think that interphase is a part of meiosis. Actually, I usually call interphase as a growth phase. Yes. Why is it growth phase? It's like when your mother gave birth to you, you didn't give birth immediately. You were given time for you to grow. So that growth time is what called the interphase in these other cells. So when it is ready to divide, it means that now interphase is ended. Therefore, there are some things which occur in the interphase, and some of them we have number one, DNA replication, which takes place. Number two, chromosome, which are single threads, become double stranded. They were single. We saw it that before DNA replication, the chromosomes look like this. After DNA replication, the chromosome become double stranded. So we ask you a question that when does DNA replication occur? We say it occurs in the interphase. That's why here we are saying that during interphase, some of the events of interphase, we see that DNA replication takes place, whereby the single-stranded chromosome become double-stranded chromosome. Uh, Someone from another chat is saying they can't wait for genetics. Yes, we will reach their genetics, and we also will try to smash it down. Yes. All right. Each chromosome will now consist of two. It will now consist of two. Consist of two what? Yes, it will consist of two threads. Yes. Or strands. It will have that and it will have that. Each strand is called a chromatid. Yes, it will have now two chromatids joined together by the center. This center is what you call the centromere. Yes. So basically, that is what you call um, interphase. So we saw that DNA replication occurs, and the function of DNA replication is to double the chromosome number or to double the genetic material. Why is genetic material be doubled? So that it can be shared equally during cell division. Yes, it should be shared equally during cell division. Yeah. So, what are some of the phases of meiosis? These phases are abbreviated as IPMET, IPMET, whereby this I is not phase of meiosis. So here we have prophase, metaphase, anaphase, telophase, of which when you say IPMAT, if you say IPMET, IPMET, whereby this is interphase, then it's prophase, this is metaphase, this is anaphase, and then this is telophase. Now, if you are saying this interphase in actual sense in English, interphase means in between. Yes. Preparation, this is preparation, this is middle, yes, and then this is apart, and then this is the terminal. So basically it still gives you as IPMAT, interface, in between, preparation, middle, apart, and yes. So basically that is it. So now when you come back here, we shall see that these are the phases which we're going to look at. So we said that interphase is not part of meiosis, so you need to know that. So when you are going to do some questions, you'll be able to understand this better. So meiosis has what you call the fast meiotic division. Sometimes we call it reduction division. Why do we call it reduction division? Because when I start with the 10, yes, I end up having two cells which have 5, 5. So the number of chromosomes here are reduced. Ne? Reduced. That's why you call it reduction division. While meiosis 2, the number of chromosomes will remain 5, 5. I started with 5, I ended up with 5. So it's not reduction division. Yes, it's not reduction division. Yes. So the diploid cell become haploid. So what happens during interphase during prophase one? The chromatin network will condense. Condense means shortening. They become visible. You are able to see why, because they have shortened. Yes. And then when they become shortened, the homologous chrom. Now look, at first it was chromatin network. When it shortens, we can see now the chromosomes. We can see the chromosomes. It doesn't stop there during interphase. The one which look alike, they will come together. This and this, this and this. So they will be like this. Therefore, I will say that the homologous chromosomes are formed. That's what you are trying to say here, that the homologous chromosomes, they are formed and they come together. They condense, they shorten. The moment they shorten, you can see the chromosomes. The chromosome become visible, and then the ones become visible, the one which look alike, they come together. That's why he is saying that when the homologous chromosomes are formed, crossing over takes place. So basically here, what happens is one chromosome and another chromosome, they cross over at a point called chiasma. So chiasma, this is the point, and then what happens there, we are able to have what called crossing over. When crossing over takes place, you experience exchange of genetic material. This is what I'm saying. This is where crossing over takes place. Please don't confuse chiasma with centromere. This is a centromere, this is a centriole, this is a spindle fiber, this is a centriole, this is a centromere, and the whole chromosome is called homologous chromosome. Sometimes we call it bivalent. Bivalent. Yes. So I think I'm clear there. If you have a question, you let me know, because this class is live, so that we are able to explain that concept you are looking for, or you want me to help you with out. All right. Then we have, after that, now the nuclear membrane starts to disintegrate. It starts to break down. That's why we're saying that the nuclear membrane disappear. It starts to disintegrate. Disintegration means it starts to break down. Yes. And then now the process of crossing over has to take place. Yes. I'll explain the process of crossing over just in few minutes. All right. After that, now the cell will undergo what called metaphase. When you say metaphase, chromosomes are in the middle. Look, you have a cell, you have the you have the spindle fibers, they are formed there. Yes, they are formed there. Now the chromosomes, they come to the spindle fibers in the center of the cell. See, in the center of the cell. They are align themselves in pairs, in homologous pairs. And then they align in the equator. Equator means the center of the cell. Yes. How do they align here? They do that randomly. That's why here, when you come here, we say that homologous chromosome randomly. This word is a key point. If you don't mention it, you get it wrong. You lose a mark. They randomly line up. They randomly line up where exactly? At the equator of the cell in two rows, meaning that if you look at here, you see that this is the first row, this is the second row. Yes. So how do they align themselves there? They are being attached by they're being attached by the spindle fibers, or sometimes we call it spindle threads. Yes. So now basically that is what is happening there. From there, we experience the next phase. We experience the next phase of anaphase. Now, the chromosomes, once they align themselves along the equator in two rows, and they align the homologous one, they are supposed to be together. They align in two rows on the equator randomly. They have to do that randomly. Not that this one is a must that you're supposed to be here, this one is supposed to be here. No, it happens randomly. Once it happens randomly, then automatically it undergoes the next phase, which we call the anaphase. In anaphase, now these spindle fibers, they're supposed to shorten. When they shorten, yes, what happens? The chromosomes, they start to move to opposite poles. You understand? So they split. You see, this one goes this side, that one goes that side, this one goes this side, that one goes that side. That's why we are saying here that the spindle thread shortened and it pulled the homologous chromosome to opposite poles. Check, it was a homologous chromosome here. It was a pair. Ne? So now they are being put to opposite poles. Chromosomes of the homologous chromosome, they are the pair, they start to separate. So that's it. That's what happens under anaphase. Then once they move to opposite poles, they reach the poles. When they reach the poles, we shall say that telophase is starting. Therefore, we shall say that chromosome reach at the pole of the cell. The cytoplasm and the nuclear and the membrane, they start to divide. This division, the division of the cytoplasm, in science we call it cytokinesis. Cytokinesis. Yes. So it means that now I started with the 20. Now each is gonna be 10. It's gonna be 10. Check here. We started with 4 cells. 1, 2, 3, 4. But now each cell now has 2 cells. That's why meiosis 1 is regarded as a reduction division. Yeah, that's what it means. So the nucleus reform, the nuclear membrane forms, so everything will start to form until it undergoes meiosis II. So what happens in meiosis II? It is a simple thing. What happens in meiosis II? What happens in meiosis II is a simple thing. Number one, the prophase, you got undergo prophase 2 now. The same phases, but now prophase is gonna be 2, metaphase is gonna be 2, anaphase is gonna be 2, telophase is gonna be 2. Yes. So the moment prophase 2 happens, the nucleolus also happens to disappear with its nucleus or nuclear membrane. Yes. The spindle fiber, they start to form again. The chromosome also start to be visible. And then now the centrioles, they start to move to opposite poles. You see, that's exactly what happens. But now, here, no crossing over. Please don't mix this. In prophase 2, there is no crossing over. So that's why you're saying that meiosis 2 looks just almost like mitosis. So the moment they move to opposite poles, now they have to reach the the center. When they reach the center, guys, whenever they bring a question, make sure that you look at this question in this format. Look at it. So this thing is supposed to be on the side, on your sides. That's how you see. Now you see they're in the middle. If you leave it the other way, you won't be able to see it clearly. So it's supposed to be in that version. It's supposed to be in that version. If they didn't tilt it or they didn't turn it, make sure that you turn the paper so that you see it clearly. So that's how it is. Yes. So yes. So they align themselves along the equator, but in this case, they don't align themselves in pairs. They align themselves singly. That's why here we shall say that the individual chromosome, individual meaning that one, or single chromosomes line up at the equator of each cell with the centromere attached to the spindle. It means that it aligns itself at the equator singly, yes, or individually, You understand? They align singly, yes. So you have to know that concept, ne? That those things they align at the equator individually. Yes. After that, once they align along the equator, now it undergoes anaphase. So now this thing, they start to separate. Now, it's not chromosome. At this time, we call it chromatid. We call it what? Chromatid. Yes. So they're saying that spindle fibers attach, start to contract, and the centrioles split. The centrioles, when they split, the homologous chromosome or chromatid are pulled to opposite poles of each cell. So the moment they start to be pulled, what happens? The chromatids, they move to opposite poles. Sometimes we call them homologous chromosomes. Remember, they were daughter chromosomes. Yeah, daughter chromosomes, or we can call them because at unreplicated chromosomes. But at this time, do not use that word frequently, because at this time we prefer calling them chromatids. Chromatids. Ne? Please call them chromatids. It's better to use the word chromatids. So the chromatids, they are put opposite poles of each cell. The moment they reach the pole, you see, they look like chromatids still, but they are no longer called chromatids. Once they reach the poles, we call them single-stranded chromosomes. Yes. So now they are called single-stranded chromosomes. So once they reach the poles, we call them single-stranded chromosomes. Yes. And then now, the nuclear membrane reappear, the cell membrane divide with the cytoplasm, which called the cytokinesis. Yeah, the number of chromosomes is maintained, whereby you started with the 4, you went to 2 in meiosis 1. In meiosis 2, the 2 are kept, the number is kept. Yes, that's why meiosis 2 is not called reduction division. No. So the daughter cell of chromosome, the daughter chromosome reach the poles, and the new nucleus reform, meaning that now the nucleus is gonna start to form. You understand? The cell membrane of each cell constrict. We call it cytokinesis. We saw it. The 4 haploid daughter cells are formed. Each daughter cell has half the chromosome number to the parent. Remember, we started with the 4 chromosomes, but now here I have 2. Yes. The daughter cells are genetically different. Why are they genetically different? It's because of the variations which took place. We're going to look at variation in detail um, because of the crossing over which took place. So what are some of the importance of meiosis? We saw it at the beginning that meiosis results in the formation of haploid gametes. Number 2, meiosis results in maintaining the chromosome number from generation to generation by introducing the halving effect, which the halving effect is meiosis 1, the reduction division, which outcompete the doubling effect which of fertilization. Then lastly, meiosis is also very important in bringing about genetic variation. Why is it that you don't resemble your brother? You don't resemble your sister? Yes, it's because of that. All right. So why is meiosis very important for survival? Meiosis is very important for survival because of it brings about differences in among the organisms, which you call the variation. Imagine everyone is the same. People wouldn't have been with the desires. Yes, people wouldn't have been with desires. So if they have desires, then it means that they're different. You'll find out that someone said that, when I get married, I want the one with a big bum, I want the one with the big breast, I want the one with the dark skin, I want the one with the light skin, I want the one with the, you understand? So it is not what? It is not the same. You can find that every shop is selling meat. But when you go to Chicken Licken, the chicken from there is different from KFC. When you go to maybe you call it hungry lion. The difference is also different. Yes. So basically, that's how it is. So the difference among the organisms, which called a variation, is meant to be we're able to get desire for that. So meiosis brings about that variation. Yeah. Someone from another chat is asking, in meiosis. Okay. They're saying that in meiosis one, they were in pairs. In meiosis two, they are single. Yes, in meiosis one, they were in pairs, and now in meiosis two, they are single. Yes. Star, star moon. I think I've answered you. Yes, in meiosis one, the chromosomes are in pair. We're gonna see it when you go to the question. You will see how this question can be answered. Yes. So, when we come back for the next class, we will talk about variation. But now, let's go for questions so that at least we know that our class, we are able to answer those questions we have covered today. All right. Last time, I think we saw this question. Yeah, let's look at this question. Hmm. When they give you questions, guys, make sure that um, you are able to label each and everything which is shown. For example, this is cell membrane. This is chromatid. This is spindle, spindle fiber or thread. This is D is centriole, centriole. E the cytoplasm. Yeah, because this is the cell membrane. So meaning that this one is the cell membrane. Yeah, identify the phase of meiosis. Identify the phase represented in diagram number one. Diagram number one is diagram number one. You see the chromosomes are in the middle, and then they are held by spindle fibers. So basically, this one becomes metaphase. So it is metaphase. But now, is it metaphase one or metaphase two? Chromosomes are still in double. Therefore, it becomes metaphase one. They have not separated. They have not what? Separated. Then diagram three. Diagram three is telophase. Is telophase. Is telophase one. The chromosomes have reached the poles. The nuclear membrane has formed. So it's telophase one. So this is the telophase one. Yes. And then they are saying that give the letter only of the part that the letter of the part that identify okay, it's the same question. Identify. Give the letter of the part that contains DNA. Contains DNA. The letter of the part which contains DNA. In this case, it is B. Yes. It is what? It's B. Let's clear it up. Yes, it's B. You see? Why? Because this is the chromo. The whole thing is a chromosome, but the only one strand comes the chromatid. Yes, it is a part of chromosome. It has the genetic material there. Then they are saying that attached to the centromere of the chromosome. Attached to the centromere of a chromosome. Yes, they are looking for the part which attaches itself. So the spindle fibers are the one which attach themselves to the centromere. You see, attach itself on the centromere. So, so we are done with this. We're done with this. Forms spindle fibers. So this is the centrioles. Centrioles. We come back here and look for centrioles. Is part D. So we saw it. So the answer here becomes the answer here becomes D. Name the organ in human where meiosis occur. In humans, they said in humans, so it is the testes. Yes, so it occurs in the testes. Therefore, we can say that if they ask you in plants but male, then it would have been anthers. Anthers. What do they produce? They produce spermatozoa. And then here, what do they produce? They produce pollen, grain. Yes. Question three. Question three, they are saying that the diagram below represents two phases of meiosis in the organism. It represents two phases of meiosis in the organism. Yes. So what are you supposed to say? Before you go for anything, I told you always label. So this is the centrio. Yeah. This one is the centromere. These are the spindle, fiber, or spindle thread. Yeah. Then this is the chromosome. Chromosome. Remember, it's single stranded. I mean, is just one chromosome. So that's the chromosome. Then yeah, so identify the phase of meiosis represent diagram 1. Remember, single stranded. These are chromatids now, and there is a space in between, which means apart. So it is anaphase. Is it anaphase 1 or anaphase 2? Because they are not full chromosome. So this is the, if this was like this and this was like this, it would have been anaphase 1. But the chromosome is splitting now, so it becomes anaphase 2. Yes. Then they are saying that identify part A. We saw it, it's centrioles. Part B, we saw it is the centromere. State the what happens to structure D in the next phase of meiosis. Structure D. What happens to this structure in this phase? Basically, this is the second phase, the next phase. Let me check and see the next phase of meiosis, because this is metaphase I, metaphase I, no, metaphase II, then it's undergo to anaphase II. Therefore, this structure, the chromosome, the chromatid will move to opposite poles. Chromatids will move to opposite poles. So that's what you're supposed to say there. Then are saying that name the process during which genetic material is exchanged as shown in diagram above. What happens? What causes these chromosomes to exchange the genetic material? We'll look at crossing in detail in the next class, inshallah. All right. So we are saying that here we are saying that the process of, so it's crossing over. Yes, we call that process crossing over. State the consequences if the process mentioned above does not occur. There will be no genetic variation. No genetic no genetic variation will be there, but it will be reduced. Yeah, because there are other factors which cause genetic variation. There will be a reduced uh, genetic variation in organisms. Organisms. Yes. Yeah, there will be reduced genetic variation in organisms. Organisms will not look they will look much more similar to each other. You understand? Yes. Why? Because you have removed the causes of genetic variation. You understand? Yes. By the way, don't forget to like, to subscribe, and also share for others, so that you guys, you don't miss out. Ne? Don't forget to subscribe. You follow us. Ne? Follow us, so that whenever we post, you are able to keep yourself on the channel and the distinction. Yeah, if you are a full-time student on Thundereduc, expect a distinction. Why? Because we give you everything. Give you everything. Yeah, you just have to be with your data. Just share, just subscribe, just like. Yeah, follow. Yeah. All right. And give the number of the chromosome in a present in original parent cell. That's where the story comes from. That's where the story of meiosis starts to begin from. All right. So here you're saying that give the number of chromosome. Yes, number of chromosome. When you say number of chromosome in the parent, so we have to go back and look at this. This is meiosis II. If meiosis II has two chromosomes, yes. What about meiosis I? Remember that meiosis I reduction division, it reduced it. Therefore, meiosis I will have 2 times 2, which gives me 4. If this one has 2, remember I said that if you meiosis I has 10, meiosis II is going to have 5. You understand? Now, if meiosis I has 4, meiosis II is going to have 2. So now it means that they have given you meiosis II. They're asking you what will happen, how many chromosomes were there in meiosis I. Therefore, they are going to be 4. So the answer here is supposed to be 4. A human cell in the same phase. Now they're talking about the human cell in the same phase. When they say the human cell in the same phase, you come back here. This is meiosis II. Yes. So in this case, the human cell will have 23 chromosomes. 23 chromosomes. Remember, in meiosis I, the human cell will have 46, which will divide into 22, 23, and 23. You see? Yes. So that now in meiosis II, each will have 23, 23, 23, 23. So in meiosis II, the number of chromosomes are being maintained. They're being maintained. Yes. Then the next question, they are saying that the diagram below represent the distribution of the chromosome. The distribution of chromosome number 21, chromosome pair, yes. So, but now we have not talked about Down syndrome. Yes. Let me check why the gametes, okay, don't have, okay. Okay, we shall see. We have not talked about a non-disjunction. It means that it will be our next class. So this, let me just call it next class. After doing non-disjunction, yes, we will come back to this question and then we'll be able to answer it. Even you saying, asking you to describe the process of crossing over. Therefore, we will be able to answer all those questions in our next class tomorrow. Inshallah, God willing. Yes, by the way, inshallah means God willing. Ne? Yeah. Right. Now, we saw the karyotype last time. Yes, we saw the karyotype. So, which karyotype is this? Is this for a male or is a female? Eh? When they ask you that question, you go to chromosome number 21. Sorry, 23. If the two chromosomes are of the same size and shape, therefore we shall say that that is X and X. If they are different sizes, therefore it's going to be X and Y. And don't forget that we say that if it's X and X, it is a female, a female. If it's X and Y, it is a male, a male. Yeah. So someone, okay. I think guys, I'm clear about that. If you have any question, let me know. All right, let's go to this question now. Uh, Give the letter of A. What is A? These are homologous chromosomes. They're homologous chromosomes. Yes. Remember chromosomes which look alike. They have the same shape, size, genetic genes are having the same locus. Yes. How many autosomes are in this nucleus? Autosomes are counted from pair number 1 to pair number 22. But how many chromosomes? Remember, they are in pairs. If they are in pairs, therefore it is, they're saying how many pairs? No, they're how many chromosomes? Therefore, I can say it is 22 times 2, which is 44. But don't just say it's 44 because 44 is not the answer. You can count them. Look at pair number 21. They are not two, they are three. Therefore, I'll add one because it's supposed to be two, but it has an extra chromosome. Therefore, it's gonna be 45. So the answer here will be 45. If you want to count, you can count them from here to there. Pair number 22. So don't rush. Don't rush to give the answers, because if you rush to give the answers, you'll end up getting wrong things. Yes. Then they're saying name the type of autosome of chromosome at position number 23. So we call them gonosomes. Call them what? Gonosomes. So we saw these from our book, from the distinction material, when I was explaining everything regarding to that. Yes, in our previous class. You see, yeah, we call them what? Gonosomes. Make sure that you get that distinction material so that you are able to score yourself a distinction. They are saying that what evidence suggests that this karyotype is for a male? What evidence shows that? If you look at this, is for a male, is chromosome number, the pair number 23. The two chromosomes are not of the same size and shape. See, which shows that one is X, one is Y. Remember, we say that here that X is longer and bigger over Y. When you come back here, you see it's longer and bigger over Y. Therefore, this is X and then this is Y. Suggest the evidence, okay? Name the type of mutation represented in the diagram above. We have not gone to non-disjunction, but this type of mutation, we call it chromosomal aberration or chromosomal mutation. Yes, mutation which occurs on the chromosome. If it was on the gene, it would have been gene mutation. But this one is the whole chromosome being added. Therefore, we call it chromosomal aberration. Then they are saying, oh, chromosomal mutation. Describe the event in Down syndrome. I'm not going to explain that. Why? Because we are going to discuss about it in our next class. So please do not miss out our next class. Make sure that you are always here so that you obtain a better understanding of this life science. Yes. Let's go to this question so that when we come back, we'll talk about uh, those other questions. They're saying that identify X. X is the centromere, and then Y is the point of crossing over, which I mean X, which we call chiasma. Yes. So X is chiasma, Z is centromere. Give one observable reason why the chromosomes above are regarded as homologous, because they have the same shape and same size. Same shape and size. And they're saying that one process occurs that has the same importance as crossing over. What does crossing over do? It brings about variation. So they're just asking you other factors which cause variation. Random arrangement of chromosomes during metaphase is also another. Yes. Then they're saying if the mouse egg contain 20 chromosomes. Remember they're saying egg, of which this is a gamete. And gamete are haploids. So how many chromosomes will there be in the skin cell, which is a somatic? Skin is somatic cell, which is diploid. You see? Therefore, I will just multiply it by 2. So 20 times 2, yes, so that I get 40. So the answer here is going to be 40 chromosomes. That's it. And then you get your marks. It's not that difficult. You just have to understand and then try to apply. Then they're saying here that name the phase of meiosis during which process represented above takes place. Yeah, this is crossing over, whereby we shall say that this is the prophase. If you say prophase just like that, we cross it. Prophase I. It occurs in prophase I. Describe the process of crossing over. We will describe it in the next class. And then they're saying explain the importance of crossing over brings about genetic variation. Yes. Do you resemble your father? Do you resemble your mother? Yeah, I always ask you a question. If you don't resemble your father and you don't resemble your mother, how sure are you? It's not personal, right? Yeah, it's just a question. How sure are you that your father is your father? You don't resemble him. Maybe you resemble your neighbor. Why do you resemble someone else in your family, but you don't resemble your father and mother who gave rise to you? We'll talk about it when we are describing crossing over. So basically, crossing over is very important in bringing about genetic variation. So we'll discuss this question in our next class. Yeah, so our next class will be crossing over, and then after that, we'll talk about the non-disjunction, the mutations and the variations which occur in meiosis. Stay tuned. Tomorrow, inshallah, we will talk about it. Don't forget to subscribe, to like, and share to others. Make sure that you share to them so that you don't miss out. See you again in the next class.